Hello viewers, today we are going to discuss about citric acid cycle, which is also called as tricarboxylic acid cycle, Krebs cycle, citric acid cycle, etc. The all these different names with the same pathway but having different names. So let us begin with its definition and where it takes place and how it takes place. Let us discuss. Definition: Tca cycle is final common metabolic pathway for oxidation of acetyl-CoA obtained from carbohydrates, lipids, and proteins to carbon dioxide, water, and energy. It means the major biomolecules, carbohydrate, lipids, proteins, which are going to enter metabolic pathway, will have common metabolic pathways as citric acid cycle, in which the products like carbon dioxide, water, and energy is produced. In this, we can see the depiction. Glucose, which is obtained from carbohydrate, is converted to pyruvate, and the glycogenic amino acids obtained from proteins, they are converted to pyruvate by various reactions. This pyruvate from carbohydrate or from amino acids converted to acetyl CoA during oxidation or in presence of oxygen, and the ketogenic amino acid are also converted to acetyl CoA or fatty acid are also converted to acetyl CoA. In this, you can see the carbohydrate glucose and then the proteins glucogenic and ketogenic amino acids entering through either pyruvate or acetyl CoA by ketogenic amino acid and fatty acid by beta oxidation converting to acetyl CoA. This acetyl CoA obtained from the carbohydrate, proteins, and lipids enters Krebs cycle or Tca cycle. Tca cycle is the central pathway connecting almost all individual pathways, either directly or indirectly. Hence, all the three major biomolecules enters Tca cycle. Next is where this Tca cycle takes place. Tissue site. All the tissues of the body except RBC and lens, you can see Tca cycle occurring, because in RBC and lens you don't have mitochondria, and TCA or Krebs cycle takes place only in mitochondrial matrix. Next, you can see intracellular site mitochondrial matrix. This mitochondria is not present in RBC and lens cells. Hence, we cannot observe TCA cycle in these. Next is starting material. The Krebs cycle or TCA cycle starts with acetyl CoA starting material, and end product is carbon dioxide, water, and energy. So let us see how this starting material acetyl CoA pass through the TCA cycle, and then how it is going to liberate carbon dioxide, water, and energy. The detailed citric acid cycle is shown here. In this, you can go through the starting compound acetyl CoA. Acetyl CoA is converted to citrate with the help of oxaloacetate and release of coenzyme A with addition of water in presence of citrate synthase. Like this, the whole cycle pass through several intermediates with various metabolites and coenzymes involved along with the enzymes, and reproducing the starting compound oxaloacetate. How it takes place step by step, and what are the intermediates and what are the byproducts or coenzymes involved, let us discuss. This is having 10 steps, so as a whole I will go it in a rapid manner, and then we'll study in slow manner. Oxaloacetate combines with acetyl CoA to form citrate. In presence of citrate synthase, water is added, coenzyme is released. Citrate is converted to cis-aconitate in presence of water. In presence of enzyme aconitase, cis-aconitate is converted to isocitrate in presence of water and enzyme aconitase. Isocitrate is converted to oxalosuccinate in presence of isocitrate dehydrogenase. NAD is converted to NADH plus H plus. Oxalosuccinate is converted to alpha ketoglutarate in presence of enzyme isocitrate dehydrogenase with liberation of carbon dioxide. Acetyl CoA is having two carbon and one of the carbon is removed in this area, you can see, and containing alpha ketoglutarate is converted to succinyl CoA. Here NAD is converted to NADH2 and one more carbon dioxide is released. So this is the second carbon dioxide of acetyl CoA carbon released in the form of carbon dioxide. And in presence of enzyme dehydrogenase complex, succinyl CoA is converted to succinate. In presence of succinate thiokinase, GTP is converted to GTP and coenzyme is released. And then succinate is converted to fumarate. In presence of enzyme succinate dehydrogenase, FAD is converted to FADH2, fumarate is converted to malate. In presence of enzyme fumarase, in presence of water, malate is converted to the starting compound oxaloacetate. NAD is converted to NADH2 in presence of enzyme malate dehydrogenase. This is rapid way of presentation. Let us see in slow manner, one by one, step by step. This is the first step, oxaloacetate, which is present in the mitochondrial matrix. It is going to combine with acetyl CoA obtained from the carbohydrate, proteins and lipids. And this acetyl CoA in mitochondrial matrix combine with oxaloacetate to form citrate. Oxaloacetate is 4 carbon, acetyl CoA is 2 carbon. So 4 plus 2, you will get 6 carbon compound, that is citrate. In presence of enzyme citrate synthase, water is added, and you can see in acetyl CoA starting. Acetyl, after the acetyl, the word CoA is there. In the next compound citrate, there is no name CoA. That means CoA is released or removed. Hence, you can see CoA is removed. CoA is a coenzyme obtained from vitamin. Citrate, next second step, citrate is converted to cis aconitate in presence of water, in presence of enzyme aconitase. In step number 3, cis aconitate is converted to isocitrate in with liberation with addition of water in presence of enzyme aconitase. So this is the first 3 steps of Krebs cycle: oxaloacetate to citrate, citrate to cis aconitate, and cis aconitate to isocitrate. In the second and third step, the reaction is performed by same enzyme. Continuation, isocitrate is converted to oxalosuccinate. NAD is converted to NADH plus H plus in presence of enzyme isocitrate dehydrogenase. It is easy to remember because you can see in isocitrate to oxalosuccinate the reaction is oxidation reaction. That means oxidation means addition of oxygen or removal of hydrogen. In this case it is removal of hydrogen. Before the arrow mark you can see NAD. After below that you can see NADH. That means after D, which doesn't have an H, after the reaction you can see D is having H. That means the H is obtained from isocitrate or H is removed from isocitrate. Removed means deleted, delete means D. So if you observe the enzyme name dehydrogenase, D means delete, removed. D only 2 letters is taken, hydrogen is removed, so dehydrogenase. And from which compound it is released or removed? Isocitrate. So enzyme name is isocitrate dehydrogenase. The product obtained is oxalosuccinate. This is the fourth step. Next is fifth step. Oxalosuccinate is converted to alpha ketoglutarate. In this oxalosuccinate, which is a 6 carbon compound, it is going to release one carbon in the form of carbon dioxide and to form alpha ketoglutarate. This reaction is performed by the same enzyme isocitrate dehydrogenase, which is a multifunctional, means it can perform several type of functions and even removal of carbon dioxide. Next is sixth step. Alpha ketoglutarate is converted to succinyl CoA. In this, coenzyme is added. Since CoA is added, the next product contains name CoA, so you can see in the next compound name succinyl CoA. And in this 5 carbon atom for alpha ketoglutarate, one carbon is removed, so that is going to form 4 carbon compound succinyl CoA. NAD is converted to NADH. Before the reaction NAD, after D there is no H. After the reaction you can see next to the D you can see H. That means hydrogens are removed from alpha ketoglutarate. Hydrogen is removed means dehydrogenase. Delete the hydrogen, dehydrogenase. And if from which compound it is released or removed, it is removed from alpha ketoglutarate, hence it is called as alpha ketoglutarate dehydrogenase. Next is succinyl CoA. Succinyl CoA is converted to succinate and GDP is converted to GTP. In succinyl CoA, next to the succinyl CoA is there. Next to, after the reaction, you can see only succinate. That means CoA is not available. That means it is removed. And this reaction is performed by succinate thiokinase. This NADH and GTP are all energy molecules. So in this depiction is done in orange box to show that they are energy molecules, so that we can at the end for bioenergetics we can calculate these steps, energy molecules for energy production or calculation. Next, succinate is converted to fumarate. FAD is converted to FADH2. So from succinate hydrogen is removed, that hydrogen is attached to FAD and it is converted to FADH2. And in presence of enzyme succinate dehydrogenase. Since hydrogen is removed from succinate, that's why removal of hydrogen dehydrogenase, and it is removed from succinate dehydrogenase. Fumarate is converted to malate in presence of water, in presence of enzyme fumarase. And malate is converted back to oxaloacetate. NAD is converted to NADH plus H plus in presence of enzyme malate dehydrogenase. From malate hydrogen is removed, so enzyme name is dehydrogenase. And since it is removed from malate, the enzyme name is malate dehydrogenase. Here also in this slide you can see orange boxes, that means it indicates energy molecules. FAD is your index finger where you are having 3 digits. NAD is equivalent to 3 ATPs. FAD you can see after succinate to fumarate, in that FAD is there. That FAD is like your thumb where you are having only 2 digits. That means FAD gives only 2 ATPs whereas NAD gives 3 ATPs. This is the step-by-step process of citric acid studied in detail. So in this you can see this is the detailed explanation from first slide of the citric acid cycle. In this slide you can see NADH between associated oxalosuccinate. In this you can see alpha ketone to succinyl CoA, NADH is there, and succinyl CoA to succinate GTP is there. And in here succinate to fumarate you can see FADH, and after that you can see next to malate to oxaloacetate you can see NADH. So these you should remember for calculation of energy. Now the topic is energetics of TCR Krebs cycle. So whatever just now the slides I showed, that steps are written here. Enzyme names with the coenzymes or energy molecule. Isocitrate dehydrogenase, the coenzyme is NADH. One NADH, as I told you, if you see your index finger is having the 3 digit, that means one NADH means 3 ATPs. And alpha ketone dehydrogenase, it also produces NADH, here also 3 ATPs. Succinate thiokinase produces GTP, only 1 GTP. 1 GTP is equal to 1 ATP, hence 1 ATP. Succinate dehydrogenase, which produces FADH2. FADH2, as I told, it is like thumb, which you are having through 2 digits equivalent to 2 ATPs. And then malate dehydrogenase, it produces NADH. NADH is equivalent to 3 ATP. So all the steps where NADH, FADH or GTP energy molecules only is calculated. At the end you can calculate 3 plus 3 6 plus 1 7, 7 plus 2 9, 9 plus 3 12. Total 12 ATPs are produced for one acetyl CoA. For one acetyl CoA 12 ATPs are produced. So one molecule of acetyl CoA gives 12 ATPs in TCA cycle. Glycolysis gives 2 pyruvate, 2 pyruvate gives 2 acetyl CoA. Similarly in lipids for beta oxidation, depending upon the type of fatty acid you will get, and according to the number of carbon present in a fatty acid you will get number of acetyl CoA in it. So this is about energetics or bioenergetics of TCA cycle, showing 12 ATPs production for one acetyl CoA entering TCR Krebs cycle. This is importance or significance or function of TCA cycle. The primary function of TCA cycle is the provision of energy. So main function of TCA is to provide energy. It gives lots of energy. One acetyl CoA gives 12 ATPs. And the TCA cycle is an amphipolic pathway. Amphipolic means like amphibians. Amphibians means which can live on land and also in water. Similarly, in this means amphipolic means they are performing both a catabolic breakdown and anabolic synthesis reaction. So glycolysis is only catabolic pathway, but Tca cycle is it's both catabolic and anabolic, hence it is called as anabolic pathway. Catabolic role. Tca is final common metabolic pathway for production of energy. The acetyl CoA obtained from carbohydrates, lipids and proteins, they are going to get converted to energy compounds such as NADH, FADH and GTP as we just discussed now. Next is the anabolic role. Starting from the intermediates of Tca cycle, a number of compounds can be produced in the body. So various type of intermediates of Krebs cycle are used for the production of various type of other compounds. In that first one you can see, we'll discuss one by one which are the compounds synthesized. One is heme synthesis. Succinyl CoA, which is produced during the Tca cycle, is used for hemoglobin synthesis. Second is synthesis of aspartate and glutamate. So certain amino acids are also synthesized, and oxaloacetate and alpha butyrate can be converted to amino acid such as aspartate and glutamate respectively by transamination reaction or process. Next is certain intermediates are utilized for fatty acid synthesis. Citrate can be transported out of mitochondria into cytoplasm, where it can get back acetyl CoA to acetate and then use it for fatty acid synthesis. And glucose synthesis. Intermediates of Tca cycle can be a source of gluconeogenesis. Non-carbohydrate sources are utilized for synthesis of glucose by gluconeogenesis, and in that Tca intermediates are also involved. This is about the importance of Tca cycle. So we have discussed the definition, tissue site, intracellular site, and then starting and end product of Tca cycle and the complete cycle in detail we have discussed along with energetics and function at the end. Thank you.